first one is called. Well, this is just a filler slide that I'll get to at the end if we have time. And that's 1 trillion bacterial cells in a liter of E. coli, and has a computing power of about a laptop. So I'll talk about synthetic biology at the end. Milkshake. So first off, the name. The name comes from the fact that we use a modified maltose binding protein, and so it's maltose malt shakes, milkshake. In milkshake, what we do is we take a maltose binding protein, MBD, and we've modified it to have a sortase acceptor site on it. So sortase is a peptidoligase. It recognizes a specific sequence, and then it'll glue anything with a glycine onto it. So it's again, it's a peptidoligase. And so what we do in milkshake to validate antibodies against post-translationally modified targets is we make 2 peptides, one with the target on it, the modification, and the second is unmodified. Now, most PTM antibodies are generated with peptides, and so we don't really have to worry too much about conformational ambiguities in it. And so there's 2 versions of milkshake that we use. One is called vanilla milkshake, which is just the plain protein. So we have the first protein, which is just modified maltose binding protein. The second one is the modified peptide glued onto it, and the third one is the unmodified peptide glued onto it. And so in vanilla milkshake, we're just going to add these 3 purified proteins to a gel, and then use a Western analysis. So the reason that we also do Westerns is that most PTM antibodies are used in Western analysis of cell lysates. The second method that we use is called strawberry milkshake, and the strawberry comes from stringency. You okay? So strawberry milkshake comes from stringency, and so what we do there is very simply add a cell lysate, HEK cell lysate to it to see if we get more than a single band. And I'll show you some examples in the following slides. And so this is what the results should look like. If the antibody that we're testing is modification specific, then we should see the latter, should see an empty lane corresponding to the maltose binding protein by itself, the modified maltose binding protein. The lane with the modified peptide that's been glued onto the milkshake protein lighting up, and then the last one where it says 0%. Now, there's a couple of reasons that we do this. One is it's 100% modified, 100% not modified. So it's a very clear answer with it. The other thing we can do is to make sure that the peptides are actually onto the maltose binding protein is there's an HA tag at the end of the peptides, and so we can just screen it with HA to make sure that that lights up correctly. So those are those 4 lanes. This is the original maltose binding protein without the tag, and then these are 100% modified and 0% modified. So it's very high tech. We've discovered that if you go to Michael's in the US, I don't know, TK Maxx here, and buy bead organizers, that you can do Westerns in them. And so we do a lot of Westerns, and so we've decided to use this as a very fast way of doing it. And it's important that the application that we're using is somewhat identical to the application that we want the people to use, the researchers to use. So you don't want to compare a Western antibody to one that's using flow cytometer. We want to look at Westerns for Westerns, and this is the way we do it. We've also used, having said that, we also use a machine that's also made in Brantford called ProteoWise, and it's an automated Western, and we've been playing around with that to see if we can get this to work. Okay, so now I'm going to go through the what I've termed the Hall of Shame, but there's actually some castles of greatness in here. There's something called conformational bias. So I'm going to show you after hundreds of gels which ones don't work. So the take-home message is not every other antibody doesn't work. It's that the ones I'm showing you for the most part are examples of things where it doesn't work. And so here's a first example of AKT phosphoserine 473, which is the best-selling anti-PTM antibody, and I think we got the information from CiteAb, so thank you. And you can see here there's 3 different manufacturers, A, B, and C, and it's not like it's closely coded. If you asked me what it was, I would do it, but it's tough to get something published where you're doing something like this, and the publication itself is taking ad dollars from manufacturers. So there's 3 different vendors looking at the same site. You can see here in the first one, it looks great. 0 binding to MILKSHAKE, good binding to the phosphoserine 473, and no binding to 473, as opposed to these two vendors whose antibody doesn't show as much specificity as the first one. So that's an example of that. So the question then is, of these, what else is there? So with the spiked HEK cell lysate, the strawberry version for stringency, what we can do then is to look at the lysate added to each of these lanes. And in the first one, which is the same vendor, you see this band lighting up, but it actually is AKT. It's good to know that. AKT is this size, and this is the maltose binding protein. In the other two examples, though, you can see that in this one, you get another band coming up with vendor B, and vendor C, you get a high molecular weight band. So looking at this, if you're using AKT 473, you probably would want to use vendor A. So it's not to look at this and say that 66% of the antibodies we tested in this case are wrong. If you go by publication number, A is used thousands of times as compared to B and C. So Andrew's not here, so I can say I don't know if I completely agree with him that 50% of all antibodies that used are wrong. If you go by that number of 1,000 antibodies used, that is correct, as opposed to maybe 20 that are used in the other cases. Really? Okay. I thought I could tell quarter off. No. 7 minutes. Okay, all right. I asked, it doesn't matter. Okay. This is another example where we can look at two different phosphorylation sites, P202 and 204. So these are two amino acids apart. In here, you get where we've designed a peptide that has both of these phosphorylated, one of these phosphorylated, the other phosphorylated, no phosphorylation. And so you can see here, although these are all sold as being recognizing both modifications, the first one actually recognizes both modifications, and you can even sort of quantitate how it does that, whereas you can see the second one does not recognize phospho 204. And the same thing with this one, and it also has a higher molecular band. These are strawberry milkshakes, I forgot to say. And again, in looking at junk 1, where it's two amino acids apart for phosphothreonine, again, you get the same thing where none of these antibodies are really saying what they should be saying, although it's interesting that you could compare this one here, which does give you some background against 183, with this one. If you put the two together, you'd actually be able to tell the difference. Looking at methylation, so those were phosphorylation. We've looked at other types of modification. This is phosphorylation. So you can see the difference here in vanilla. And then in strawberry, you get these extra bands coming up with these two vendors. In this case here, same thing. You get these bands. And this has all been published, so I'm not going to walk you through it that way. This is a lysine, methylated lysine at this particular position. This one is interesting because this vendor sells it as recognizing lysine K9 methyl 3 trimethyl, and in this case, it only recognizes it when the phosphoserine next to it is phosphorylated, and this one is the opposite. So it's a good way to use both of those to tell you whether the phosphoserine next to it is modified or not. You can get the total amount of it in one case, this one, and in the other. This is acetylation. I will say, for some reason, acetylation, I don't think we've ever found a bad acetylation antibody. So if you're working in acetyl groups, it's nice. We've extended through a friend of ours, Brian Kay, who many of you know, looking at a different protein that's been modified called CBD. And the reason we do that now is we can put both of these in the same lane of unphosphorylated or unmodified and modified in the latter lane, and not be able to take up a spare lane. So this is a different molecular weight. We now have one that's 52 kilodaltons, another that's about 25. So they end up in an area of the ladder that's pretty clear. Unfortunately, I picked the one partner in doing this experiment who's never smoked marijuana. So I was originally going to call it weed, but Brian's that's a Boy Scout. So this is looking again at monomethylation, but this is in this case here we're looking at the commercial lysates, and it's not a fair comparison here because they're not made for these particular antibodies. I'm just saying here that the commercial lysates themselves need to be validated. This is Sundae, so MILKSHAKE and Sundae. This is 20 flavors. So at a particular site in an epitope, for example, we may look to see if, in this case here, we're looking at this Emperor sequence from HIV, this aspartic acid. We want to know what else do the antibodies that we've isolated against it recognize. So we replace the D with all 20 amino acids to give us Sundae. And then look at it against the peptide and against the full-length protein. In this example here, you can see this antibody 42 recognizes the peptide, but not the full-length protein, which is in gray. This one, 37, recognizes both, so that's good. And it's very specific. Whereas these two are somewhat they're good enough to be specific, but they also recognize other things and they have a higher background. So that's Sundae. And so I'm going to switch in the last couple of minutes. I have to quarter. Yeah. Okay. 2 minutes. Okay. Yeah. So I'm going to switch to our other. So half of the group is working on MILKSHAKE and Sundae. The other half is working on EpiVolve, which is a method that we've developed for site-directed antibodies. And so the idea is, given a protein sequence, can you get an antibody targeted to a specific amino acid in that sequence? And this is the way we do it. What we do is we first immunize the animal with an immunogen where that modification, where that position that you're interested in is modified. So, for example, if you have 100 amino acids and you want to get an antibody against position 60, we would make an immunogen where position 60 is now a modified hapten. So it's very immunogenic. It focuses the immune system in the animal to that site. It overcomes tolerance because that modification doesn't exist at that site. And it also makes any hard to target sequence immunogenic because, again, the focus is towards that particular site. And then after the rabbit has made a response, we then immunize it with the native immunogen. So really what we're doing is two things. One is priming the pump of B cells against the target sequence, and then looking for, through somatic hypermutagenesis, where the response of the B cells have mutagenized to now recognize the native sequence, and those are the ones that we're targeting through B cell isolation. We've done this now in vivo with chickens, rabbits, mice, many different strains of mice. This is phage display. It also works in that, but Andrew's not here, so I'm going to skip it. So with that, you could do, for example, a global trek around a protein, looking for spots to see. In this case, we're interested in the COVID, along with everybody else who was working in 2020, to look for spots of it where it may be viral neutralizing, but it's not usually picked up in normal screens around the ACE2 binding pocket, which is up here. So we made antibodies against all of these pink sites on it, and this is an example of one of them, which was directed against position 405, which in the original Wuhan strains of SARS was a tyrosine, but in the Omicron had mutated to a histidine. And so you can see with EpiVolve, you can actually get amino acid variant antibodies that are specific to an amino acid. So in this case, we have ones that are histidine responsive, ones that don't matter what's there, whether it's histidine or tyrosine, and others that are tyrosine specific. We can look at the splice junction sites by looking for antibodies that target just that particular neo junction, but not the other, and that's shown here. KRAS is interesting. There are the normal position is glyc12, G12, but it mutates to 6 other amino acids. This is an example where we went after one that was G12D specific. We have the other 4 or 5 in our lab, and showing you this one. Basically because it's the best, and why would I show you anything but that? If you go to a PEGS meeting, every third talk is about how GPCRs are difficult targets, but they're going to tell you how to get it, and I'm not going to do that. I'm just going to tell you that they're very tractable using EpiVolve. And so if you go after the ligand binding pocket, you can actually get ones that are agonists or antagonists at that. This just, again, KRAS showing you in different assays, Westerns, ICC, IHC, and actually the antibody I've been showing you is directed against the G12 peptide in the HLA pocket, and it works as a CAR T. And do I have a minute or? Okay, sorry. Let me go on and just thank the people who were there. Okay. Um, the two projects I talked about have been published. Epivolve's been published, and the milkshake antibody validation has been published. The people who did the work, I'm smiling, so it must have been an early picture. Kiana did the milkshake. Mary supervised a lot of the Epivolve studies. And these are the people I'd like to thank, the Avatech team, Kevin Harvey, who's not here, but many know, Brian. And then Jim, to answer your question, we didn't do this for free. We had NIH support to help us on this project. And that's it. Thank you.